Hello to everyone. Good morning, everybody. Good afternoon, good evening, wherever you're joining us from, whichever time zone you're in. And on behalf of WIDS Oxford, a very, very warm welcome to everyone. Thank you very much for joining us for this event on women in data science at Oxford. Just to let you know that this event is taking place as part of a celebration of 100 years of women at the University of Oxford. And we're absolutely delighted. We are thrilled to see such a large turnout today, both from within the university, and we have quite a few guests from organizations and institutes from overseas. So welcome to everybody once again. And I think let's kick things off. So first, I need to get through a few housekeeping things very quickly. The first of which is this event is being recorded. The recording will be available at our website after the event, which is so you can have a look at that. We will post a link for that in the chat as well. And so unless otherwise explicitly mentioned, please assume that the recording is on. Then we have to talk about cameras. So please make sure that your cameras are switched off for the duration, for the entire duration of the event. So ideally, this is something that we would have liked to do in a nice, pretty college where we would be able to meet in person, and over a cup of tea and biscuits. However, things are as they are, and we're here now. So we just so that we don't make teams feel that it cannot cope when there are 100 cameras turned on. So let's just make sure that our cameras are going to be off for the duration of the event. Thank you very much for that. And participants should be muted throughout the event, but just in case you find that you've for some reason been unmuted, do please go back on mute. There will be opportunities for interaction throughout the event, and we'll let you know about that as in when we get through the course of the day today. And then finally, if during the call, during the meeting, if you run into any sort of technical difficulties, our IT team are in the call and they will be able to help you. So if you just drop an email to wids-ndoms@ox.ac.uk and our IT team will try and help you get back. Yeah, okay, so with that, we're going to make a start. So we have quite a busy program for the morning today. We are going to begin with an introduction to women in data science. We then have a lineup of 3 stellar speakers who are going to present or share with us some of their research using data science that is going on at the University of Oxford. This will be followed by a panel discussion on the challenges and opportunities for women in data science. We will then wrap up and close the main event. Just after the main event, there will be a session on how to get involved with WIDS Oxford and plans for the upcoming year. So for those who are interested in getting involved, do stay on for that one as well. We will have 2 short breaks in between. So that's our program for today. So yeah, let's get started. And I thought what better way to start an event on data science than with a pop of data. So this is us basically. This is us today, or one way to look at us. We have more than 140 participants registered for this event. We are representing between us 50 departments and colleges from across the university. In fact, we have, except for humanities, we have all 3 divisions, medical sciences, MPLS, and social sciences present with us in the call. So quite a good spread of disciplines as we can see today. And then in terms of In terms of levels, we have an almost 50-50 split between budding and senior researchers, with a variety of levels of interest in data science, from just curious all the way to I can make a CNN in my sleep level of data science expertise. And finally, we have guests from 6 different international organizations. So welcome everybody. And it just goes to show the multidisciplinary shades really and variety of data science going on at the university as well as elsewhere. Okay, so with that, let me begin with an introduction to women in data science. So my name is Sarah Khalid. I am a university research lecturer and a senior research associate in biomedical data science at Oxford. And I am also an ambassador for women in data science at the university. So in a way, the women in data science, it goes back to the issue of the underrepresentation of women in STEM, really. And this is something that many of us are well aware of. It's well recognized. It's talked about a lot. And these are data from the US showing the trends over time. So the last 50 years or so and what's happened in the main sort of STEM subjects, especially in computer science. And there's a lot of theories almost about, how this has come about and so on. Well, we won't delve into that. This is just to show the overall, picture. And here's some more recent data from the UK, also depicting a similar picture in that, there's a gap, there is a gender gap, and the fact that it's not just limited to the education, but it continues all the way into the workplace. And again, this is not something that's region specific. So all over the world, actually, to varying extents, to varying degrees, we see women dropping out at various stages of their career. And the same is true, if not even more so, in the case of women in color, in terms of proportional representation and promotion in the workplace. And so it's not really any surprise then, again, as many of us know already, that less than a third of women make it at the top at the decision-making level. And this data-driven decision-making, which is arguably the decision-making of the future. So it's all the more important for the equity and diversity to be present at that level as well. Now, of course, these are gaps that are well known. There are still underlying biases, and therefore you see, unfortunately, still these kinds of scissors and leaky pipelines. But things are changing, things are moving along, and we know diversity promotes intellectual strength to solve complex problems, to do breakthrough science. Women have and women continue to achieve great heights in the world of science and discovery. There's a lot of tremendous amount of terrific work being done by women in the field of data science, of STEM. And at this point, I felt that after yesterday's news, I just had to add this this one here. And I'm sure that I speak for all of us when I say this is a moment of pride, I think, for all of us in the world of research, really, not just at Oxford, but specifically also for women in science, women in STEM. And that is exactly why and what for Women in Data Science as a network, as a group, was created, really to celebrate the achievements of women in STEM and data science, to own the narrative and to make sure that it is moved forward. So today's event, it marks the launch of Women in Data Science, the Women in Data Science Network at the University of Oxford, which will work closely with other networks in the university and beyond. It is for researchers involved in or interested in data science, students, postdocs, professors, men and women. So it's open for all, really. The idea, though, the vision is it's to inspire more women to join data science and STEM, and to educate everyone, regardless of gender. Finally, to support women in the field. So the focus is women, the network is open for all. WIDS Oxford is affiliated with and supported by the Global Women in Data Science Initiative. Now, this is something, it's a network that's spread over more than 60 countries. And they have many regional branches, all with the common aim of inspiring and supporting women in STEM, whilst educating everyone cutting edge data science in industry and academia. Across the network, there are many regional events held every year, and the highlight of the year is an annual conference which is held at Stanford University. And that's Stanford is where the WIDS initiative began around 5 years ago now. So we're very excited to be a part of this, this global movement, really. And so with that, let me briefly introduce you to the WIDS Oxford organizing team, who have worked really hard to bring this event. And we're all on the call today. We are really friendly bunch. And there will be an opportunity to talk to the team after the event if you want to stay back and talk more. We will be able to do that. So that's by way of a brief introduction to the idea of women in data science. Once again, we are delighted to have everybody join us for this inaugural launch event of Women in Data Science. And with that, I guess let me move on to what you're most interested in, which is our speaker session. So we're going to move straight now into our speaker session, where we have 3 very accomplished women researchers showcasing some of their research going on at the university and in collaboration with other institutions using data science. So without further ado, let me introduce and let us welcome Professor Naomi Allen. And Naomi is the Chief Scientist for the UK Biobank, which she will tell you all about in a minute. And she is a Professor of Epidemiology at the Nuffield Department of Population Health at the University of Oxford. Her research focuses on the use of big data to understand the role of diet and hormones on the risk of cancer. Naomi also advises governments on use of data for public health research by scientists all over the world. She will speak on her work on data science and epidemiology using the UK Biobank. Naomi, it's over to you. When you're ready, please share your slides.
Many thanks, Sarah.
Pleasure.
So many thanks to everybody who've joined this talk. I'd like to spend a little bit of time talking to you about data science and epidemiology. I can't seem to slides. Hang on a second. So epidemiology has received unprecedented attention this year, and everyone, it seems to me, has become an armchair expert about infection transmission rates, and even my kids know what an R number is. But whilst the pandemic this year has certainly put epidemiology in the global spotlight, the science of epidemiology is so much more than looking at the transmission of a virus. It really encompasses the scientific methodology that underpins public health. So the definition of epidemiology is the study of the distribution and the determinants of disease frequency in human or other populations. So we study the natural history of the disease, we investigate the potential causes or what we call the risk factors for disease, with the ultimate aim, of course, of informing public health policy to help preventing disease happening in the future, and also to help develop new treatments for disease. But epidemiology has really changed at a fantastic rate, certainly over the last 40 years or so. So at the moment, the most common causes of death are from non-communicable diseases, so heart disease, stroke, cancer, dementia. But what we're starting to see, certainly in the last 20, 30 years, is an emergence of new infectious diseases. So if you think of HIV/AIDS, SARS, MERS, swine flu, and also COVID-19, of course, these new infectious diseases often have a global spread, as we've seen with COVID-19, often with devastating impacts on the population's public health. So epidemiologists are starting to have to reassess the priorities of their research agendas to cope with both non-communicable disease and also the emergence of new infectious diseases, which is, of course, what we're dealing with this year. Epidemiology is also crucial to understanding how best to implement and evaluate preventative interventions in the general population. So if you think of things like cancer screening programs in this country, the use of medications such as aspirin and statins to prevent heart disease rather than treat disease, down to government recommendations of eating 5 portions of fruit and veg a day, all of these recommendations and interventions implemented by the government are underpinned by robust science that comes from large-scale epidemiological studies. So for non-communicable diseases like heart disease, cancer, and so on, they're caused by a multitude of genetic and lifestyle factors, each of which on their own have a small effect on the development of disease. And so you need very large studies of hundreds of thousands of participants in order to robustly identify any individual effect on a given impact of development of future disease risk. And there's also just been a fantastic advances in technology. So just the sheer amount and breadth of data that we can analyze is just like nothing we've ever had, even compared with 10 years ago. So now we have huge data on genomics at scale. We can measure thousands of circulating metabolites and proteins in our bloodstream and other biomarkers in order for us to understand the reasons why some people go on to develop certain diseases and some people don't. And in parallel with that, there's also been fantastic advances in terms of compute and storage power and data analytics in order for us to better understand and interpret how genomics, metabolites, other biomarkers affect the causes of disease. And in parallel with that, there's also has to be advances in the capability of data protection and so on, so that we make sure that we're analysing these data in the most secure way possible. Which leads me nicely onto UK Biobank, that is a large-scale epidemiological study, which I've had the privilege to be involved in with some time, that is following the health of 500,000 people in the UK to better understand how lifestyle, environment and genetic factors interact with each other to cause diseases in some people, but not in others. It's arguably the most accessible, in-depth, the largest, most easy to use and most flexible biomedical data resource in the world. And our sole aim is to enable the worldwide research community to make scientific discoveries to improve human health. Our main funders are the Wellcome Trust and the MRC, although we also receive some funding from local governments and charities as well. So UK Biobank really does have a unique combination in the fact that we have large-scale data. So we've recruited 500,000 participants aged 40 to 69 years at recruitment between 2006 and 2010. The map on the left there shows our 22 assessment centres across the UK, and we recruited people who lived within about a 30-mile radius of each of those assessment centres. They came along for a 4-hour visit, and we collected lots of information about their lifestyle, medical history, physical measures, and so on. And so the data is highly standardised and curated. And we also have biological samples on everyone, such as blood, urine, and saliva, also on a large subset. And crucially, we have linked to everyone's electronic medical records over time, so we can find out what happens to them. And we now have over 10 years of such follow-up. But perhaps most important of all is that this data set is readily accessible to researchers worldwide to perform health research that's in the public interest for epidemiological purposes. So since recruitment ended in 2010, we've continued to collect vast amounts of data on all of our 500,000 participants. So we've done repeat assessments where we bring in a subset of individuals, repeat of their recruitment baseline assessment. We have a rolling programme of web-based questionnaires that we send out to everyone for whom we have email. And the aim of that is to collect information on health outcomes that are not well ascertained through medical records. So things like cognitive function, pain, and sleep. We've also collected objective measures of physical activity through a Fitbit type device on 100,000 individuals. We've measured baseline biochemistry measures on the full 500,000. We've collected genomic data using a genome-wide genotyping chip on all 500,000. And we're currently halfway through an imaging program to collect MRI data on 100,000 participants. So you can see we just have vast amounts of data breadth and data depth in this cohort. So when UK Biobank was set up more than 10 years ago, one of our aims was to obtain genetic data from all of our individuals. And this is important because we really need to understand the extent to which genetic factors influence future disease risk in the general population. This is so we can understand the causes of disease, and obviously leads the way to precision medicine. So this is where you can target therapy or perhaps target early detection screening programs to subgroups of the population that are more genetically predisposed to developing certain conditions. And UK Biobank is by far the largest study in the world that has such a wealth of genomic data, plus all of the lifestyle data and the biochemistry data and linkage to health records to make that possible. And I think it's true to say that the genetic data we released a few years ago is really starting to transform epidemiology. So, for example, researchers can now look at the association of genetic variants across the whole genome and how they're related with different diseases and different characteristics and traits. And these association scans are now considered routine and are now publicly available in the domain. And that's made possible through the large size of the cohort, the fact that we have standardized measures on everyone, and the fact that we have linkage to health records over time. And we're also taking this one step forward and now doing whole exome and whole genome sequencing for the full cohort. So, I mean, even 5 years ago, if you'd said to me, you know, do you think it's possible that we can do whole genome sequencing, which is measuring all of the 3 billion letters in the genome for 500,000 people, I'd have probably laughed you out of the room, because at that time we just didn't have the technology, and it was just simply way too expensive in order to do that. But here we are in 2020, and this project is actually already fully underway. So the pace of change in epidemiology is so fast that we're being able to do things that even a couple of years ago we just didn't think were logistically feasible. So I just want to show you an example of just how the genetic data is really starting to enable exciting and novel research. So this is data taken from research papers done by the Broad Institute in the US that was published a couple of years ago, and they generated genetic risk scores based on the variants across your genome. And the graph in the middle shows the genetic risk score for coronary heart disease. And what you can see is that those unlucky people who are in the top 5% of genetic risk for developing heart disease have over a 3-fold increased risk of developing that condition compared to the rest of the population. So what this means is that, you know, heart disease actually has a large genetic component, which is quite different to what we thought of a few years ago when, you know, it was largely believed that lifestyle factors like smoking, obesity, and lack of physical activity were the main drivers of heart disease. And it shows that heart disease, you know, if you're in the top 5% and you've got a 3-fold increased risk, that's equivalent to many monogenic disorders. And this paves the way for personalised medicine. So you can imagine being in a situation in the future where if we all knew what our genetic risk was of developing heart disease, and you were unlucky enough to be in that top 5%, so you had a 3-fold increased risk of developing heart disease based on your genetics, you might then be much more motivated to modify your lifestyle in order to reduce your absolute lifetime risk of developing heart disease. So this type of research is really generating huge excitement about the use of genetics in public health. So as well as genomics, UK Biobank's undertaken other large-scale initiatives to measure biomarkers in the body. So for example, we've performed a whole range of biochemistry markers in all 500,000 participants, so things like cholesterol, glucose, vitamin D, and so on, to enable researchers to better understand the mechanisms through which genetic and lifestyle factors influence disease risk. Researchers are also measuring telomere length in all the 500,000 participants. So telomeres are the caps at the end of the chromosomes, and they're really important as a proxy marker for biological aging. So having these data will start to enable really unique research into age-related conditions, and these data will be available in the first half of next year. In addition, other researchers are also starting to measure circulating metabolites in all 500,000 participants, which are enabling novel research into how genetic and lifestyle factors influence our circulating levels of metabolites, which then in turn, how do those influence disease risk? So these data for the first 120,000 participants will be available in the first half of next year, with the rest of the data coming thereafter. So these cohort-wide assays are starting to enable a really wide range of research to be performed by the global research community, and are potentially really transformative in terms of the science that they'll be able to support. So I just want to segue a little bit into the imaging project that we're halfway through in UK Biobank. So we're undertaking the largest study in the world to perform imaging in 100,000 participants. We've already scanned 50,000 of them in 4 dedicated imaging centres throughout the UK, where we invite participants along for a 4 or 5 hour visit. We conduct MRI scans of their brain, the heart, the abdomen, although it's actually from the neck to the knee. We do an ultrasound of their carotid arteries in their neck, and we do a full body DEXA scan to measure bone mineral composition and body fat composition as well. And this is to, we also plan to undertake repeat imaging in a large subset of these individuals in a few years' time, so we can assess changes in the structure and function of internal organs. So what these data will do is they will enable research into the disease pathways by looking at how the structure and function of organs both before and after a disease is diagnosed. So they'll really be able to tell researchers how diseases develop, and more crucially, whether we can identify early disease biomarkers. So for example, if you think of the brain MRI scan, by looking at functional markers of the brain, we may well be able to identify early markers of cognitive decline, which may be an important biomarker for Alzheimer's disease, for example. These imaging data that we have on 50,000 are already enabling a huge array of novel research that just simply could not be possible otherwise, and is quite rightly generating quite a stir in the media and scientific circles as to how we can start to use these MRI data for large-scale research purposes. We also want to use these imaging data to help facilitate COVID-19 research. So our aim is that we'd like to enable research to look at the impact of coronavirus on changes in internal organs. So as I've just said, we've already imaged 50,000 participants, and this was before the COVID-19 pandemic. Our plan is next year to start to send out antibody-based test kits that people can take at home, where they put a finger prick of blood onto like a pregnancy type kit. This will tell them within 15 minutes or so whether they have antibodies to coronavirus or not, so whether they have previously been infected with the virus. We will then re-invite back for a second imaging visit about 3,000 of those individuals, half of whom have got positive antibodies, so are cases, half of whom have negative antibodies, so who are controls. And this will provide the only study in the world that has data, imaging data pre and post infection. So it will enable research into the direct effect of coronavirus on changes in internal organs. So a really exciting study, hopefully that will start early next year. So UKIP Biobank is a prospective studies. So we're following up the health of individuals over many decades to find out about what diseases they develop, the disabilities, their quality of life, and so on. In order to do that across the UK, we need comprehensive, scalable, and importantly, cost-effective methods in order to link to electronic health records for all of these individuals. We're starting to do this to enable COVID-19 research. So we've rapidly made available, approximately on a monthly basis, to researchers worldwide, data on coronavirus antigen test swab data from death registries, data from hospital and inpatient admissions, including critical care, and also data from primary care records available specifically for COVID-19 research. And in the cohort as a whole, we've got just over 8,000 confirmed and suspected cases of COVID-19. And these data are enabling really unique research into the association of lifestyle, underlying health conditions, medications, and genetic factors on COVID-19 severity. So we have over 670 research groups have signed up for these data to do COVID-19 research. Over 60 papers being published so far, that increasing daily. It's hard to keep up with it all. These are just some examples of the type of research that's being produced. So, for example, people looking at ethnic disparities in hospitalization for COVID-19, whether vitamin D is related to COVID-19, and lots of papers about particular genetic variants that may increase susceptibility to severe COVID-19. So a really important and rapidly changing area of research. So one unique thing about the UKIP Biobank is it's very easy to access the data. It's available to all bona fide researchers for all types of health-related research that's in the public interest. Importantly, we have a level playing field for both academic and commercial researchers to use. There's no preferential or exclusive access. We are a charity, so researchers only have to pay for the costs of using it and not for the cost of setting it up. So we work on a cost recovery model. We do require researchers to return any results back to us so that we can all incorporate them back into the resource and then share them to the rest of the research community. And this method of engagement enables academia and industry to be engaged to benefit the wider research community as a whole. So in 2012, when we opened for business to researchers, we had just over 300 researchers registered to use the resource. They were largely from UK-based academic groups. And then at the end of last year, we had just under 6,000 registers to register to use the resource in that year alone, the vast majority from overseas. So overall, we've now got over 17,000 registers in over 70 countries. So it really is truly an international resource now. And I just want to leave you with this kind of global heat map of the types of data and the types of research that UK Biobank is enabling. And what you can see is that it's not just the obvious things like cancer and genetics and imaging. It's enabling research into Parkinson's, gout, depression, fibrosis, sarcopenia, glaucoma, menopause, all sorts of health outcomes that just would not be possible in any type of research study. So I'll leave it there, and I'm very happy to answer questions in the panel discussion. Many thanks.
Okay, brilliant. That was awesome, Naomi. That was thank you for taking us on this whistle-stop tour of the UK Biobank, and for showing us the amazing and fascinating breadth of rich data that there is. And it's also fascinating to see already the amount of COVID research that this resource has enabled. So now we have an opportunity for taking some questions for Naomi. So please, if you have any questions, do use the meeting chat on the right of the meeting to post your questions, and we will pose them to Naomi. While we're waiting for participants to think of their questions and to add their questions to the chat, I've received, I think these talks have generated a lot of pre-interest before the event as well. So we have some pre-submitted questions, Naomi. So maybe I could make a start with those whilst we wait for the participants to come in with their questions. So I think one of the questions that we received already was, what are the main challenges, or what challenges have been experienced in the fact that this is obviously a prospective data set, started some 10 years ago. So if you could just give us some idea of some of the challenges of collecting such a resource over time.
As you can imagine, there are quite a few challenges, and those challenges relate to being at such a large size. So you really need industrial scale processes in order to collect standardized data for participants across the UK. I think our biggest challenge is, well, there's two biggest challenges actually. One is when you're linking to health records for all 500,000 participants, it's navigating your way through the regulatory hurdles that you have to jump through to make sure you have access to the right data, you have the right linkage, and processing and understanding all these complex clinical data sets that come in all sorts of flavours and formats from England and Wales and Scotland differently. Just incorporating all of that into a data set that is meaningful for researchers to use is an enormous challenge. So I think that's our biggest one. And the second biggest challenge we have at the moment is just sheer data size. So when we have the full genome sequencing data, we'll have up to 15 petabytes of data in the resource, which I've been told is equivalent to about 4.5 times the entire Netflix catalogue. So even with the best internet connection in the world, it will take you 3 months to download the data set. So in order to combat that, we're now moving, instead of researchers downloading the data onto their own laptops, we're now moving to the cloud, so that we're developing an analysis platform so that researchers can do the analyses in situ. And that's absolutely the way we need to go, just because of the sheer amount of data that we now have.
Okay. Thank you. Thanks. So we have one interesting question, I think in the COVID context. It's about with the COVID antibody testing. Now there have been mixed results about the duration that antibodies will remain in the body. So do you see this? How do you see this affecting your research?
Well, funny, one thing I didn't talk about was we're just performing a serology study where we're sending out antibody kits to 20,000 individuals over 6 months specifically to see how antibody levels wane over time. And what we really want to answer is, are they stable? Do the change in the antibody levels, does it change by age? Does it change by ethnicity group? And in order to inform the length of natural immunity, which obviously has huge implications for any vaccine. And those data will be coming out in the next 6 to 8 weeks or so. So I'll be hoping to share those with researchers early next year. But there needs to be a lot more research. That at the moment the research seems to be quite mixed, but I think there's a general consensus now that antibodies to coronavirus do last at least 4 to 6 months. Whether they last up to a year, nobody yet knows, obviously, because the virus hasn't been around all that long. So we just need to wait and see just how long they last. But the data so far seems pretty hopeful.
Okay, thank you very much. I mean, there's no end to the questions that one can ask about such a rich resource. I think we'll just finish maybe with this one question also to do with our topic today. So with obviously it being a biobank, there must be a lot of computer science people hiring from people in computer science fields, and those who are not epidemiologists but more to do with data science and data analysis. Is it the case that hiring in the UK Biobank and related organizations is dominated by men, and how do you support women through the hiring process?
Yeah, that's a really good question. Actually, in UK Biobank, we have quite a large team of data analysts, and the majority of them are women. They work most of them work part-time, they do job share, and they have huge analytical skill sets. And I'd say 10 years ago, that would have been a very different situation. It would have mainly been men wanting to come into these fields. But I personally have seen a real shift in the last few years of women being really confident and capable of, having degrees and postgraduate degrees in bioinformatics, data analytics, data visualization, and they're just as up there with the men in terms of being able to dominate this field. So we're starting to see a real shift in that.
Okay, brilliant. Thank you so much. So I think we'll thank you, Naomi, for your very interesting talk, and thank you to the participants for their questions. I think we can take a short break at this point, and then come back for our next speaker at 10:55. So thank you once again, Naomi. We will see everybody back at 10:55. Thank you. Ask your questions at the end of this talk as well, so do please feel free to use the chat box for that and keep the questions coming. Okay, so with that, let me introduce you to Jenna Reps. Jenna is joining us as a guest speaker from Janssen Research and Development. She's an associate director in Observational Health and Data Analytics Group at the, sorry, just a moment. I'm just checking my microphone settings here, which should all be good. Okay, yes, so we're back on. So Jenna is an associate director in the Observational Health and Data Analytics Group at Janssen Research and Development. Jenna's areas of expertise include applying machine learning and data mining techniques to develop solutions for healthcare problems. She leads currently the Global Observational Health Data Sciences and Informatics Workgroup on patient-level prediction, with the aim of developing open source and user-friendly tools for risk estimation models using big data. So Jenna is going to share with us her work on 88 hours of COVID-19 analytics. Thank you very much, Jenna. And when you're ready, you're on.
Hi. Let me just get my slides up.
Brilliant.
So hopefully everyone can start seeing this now. So yeah, I'm going to be talking about a study-a-thon that we did at the early part of this year in the end of March. And the study-a-thon, for those who haven't heard that term before, is basically a group of time where we get a group of researchers together to hack away on different problems in healthcare and try and solve it through a collaboration known as Odyssey, which I'll be talking about. And specifically at the beginning of this year, we focused on COVID-19 to try and gain insights by applying some cool data science and seeing what we can learn about the virus. So just to give you a bit of background about myself, as it was mentioned, I work for Janssen R&D, and I tend to focus on prediction in my role. So I do a lot of prediction work and trying to come up with new methods, best practices for prediction using observational healthcare data. So I tend to focus on the observational healthcare data, which I'll be talking about in the remainder of this presentation. My background is in mathematics and computer science, so I actually have a mixture of those two skills. And I'm very active in the Odyssey collaboration. This is what we're talking about. Well, the study-a-thon was part of Odyssey. So I'll give you a bit of background on that. Now, just as a warning, I put a picture of my dog. He looks cute, but he's a very grumpy old man, and he tries to bite and bark at everyone. But I've currently distracted him with a treat, so he should be quiet for the rest of this presentation. So the first question, what is Odyssey? And if you go to odyssey.org, you have a website where there's a lot of resources. So you can have a forum where you can chat with people in the community. There's a book of Odyssey where we've talked about the best practices that we've learned so far and the way we actually run projects. But effectively, what Odyssey is, it's a group of researchers with diverse skill sets from all over the world. They're all collaborating together with the single aim of trying to improve the way we analyze these big observational healthcare data sets that we have available, like claims data in the US or EHR data around the world. So we all have this big data. We're trying to come up with open source solutions. So we put all of our code to run the analyses on a repository known as GitHub Online, which means that anyone can contribute, anyone can use the solutions. It's all free, and we encourage people to be involved and add features if they see that there's a need. So we have regular meetings throughout the year, and we were actually due to have a meeting in Oxford in the beginning of this year, in March 2020, but that got cancelled because of COVID. But we decided, let's not just get rid of the whole event. Let's try and replace it with a virtual study-a-thon, where we can actually use the skill sets that we have, we can use the data that we have, and you're going to see that Odyssey actually has a power in lots of data. And we can use this time to actually try and learn about the virus. So there were different areas that people were focusing on. Some people were looking at characterization. How do the COVID-19 patients look? Do they look similar to the flu patients? Because at this time we didn't really know. So that was one area of interest. There was people looking at potential drugs that could be used for COVID-19 and looking at the safety of those drugs. So that was kind of the causal inference research area. And then there's the prediction, which is what I was involved in, and that's what I'm going to be talking about. But there was a lot of different research questions involved in this 88 hours. Prediction was just one part of that. So before we started the study-a-thon, before we started this 88 hours of intense research, we wanted to prioritize some questions that we could focus on. And we wanted to make sure that we were focusing on prediction questions that have clinical value. So we wanted to make sure that we had prediction questions that if we solved them, they would be used, potentially by policymaking. For example, if we could come up with solutions to identify high-risk people for severe illness, that could be used to help shield those people. And we also wanted to make sure that we could answer these questions with the data we have. So we sat down and we discussed it as a group on our forum, and we came out with 3 questions of interest. The first was, what's your risk of being hospitalized if you have COVID-19? So you are a bit more severe than most. And then there were 2 other parts, that if you're hospitalized for COVID-19, what's your risk of intensive care or mortality? So if you're very severe for COVID-19. And we thought this could help with the policy making, and it would be really good if we could come up with a simple model. So we wanted to apply the large scale machine learning and get some really awesome state-of-the-art models, but we also realized that this is a problem that's affecting the whole world. If we have these complex models, then there's a barrier getting these implemented. So we also wanted to look at, can we simplify a model and have something that's very readily applied around the world? So these were the 2 aims, full model and a simple model, these different questions. We also allow people to suggest other questions of interest during the study-a-thon as well. So we weren't just restricted to these. So 88 hours to do 3 prediction models or more. How is this possible? The main thing is standardization. So Odyssey has realized to have a successful collaboration with lots of different data sets, one of the key things is a standardized data structure. So in Odyssey, we all tend to map our data to this common data model known as the OMOP common data model. And the reason for this is the example here. I have data in the US and claims data, and it will have certain tables and columns. And then if I'm collaborating with someone in Oxford, they have data, maybe a UK EHR system, and it will have different tables, different columns. And if we're collaborating with someone in Korea, they're going to have a database, again, that has very different structure. So everyone has their own different structured database. And it means that if we're doing any analysis, I have to write code for my database, Oxford has to write code for their database, Korea have to write code for their database. And it means you have to do 3 tasks for analysis. Whereas if you can map to a common data model, and the OMOP common data model is being developed such that any observational healthcare data set can be mapped, we have some standardization. And this means that now if I write code to develop a model or externally validate a model on my data set, is also applicable to any of the other data sets that have been mapped to the OMOP CDM. So rather than having to write it multiple times per database, we actually only have to do it once and we can run it. So for model development, I write code that assumes you've got the OMOP CDM data. It does the whole framework to develop the model, and I can share that code, open, put it onto GitHub, and anyone with their data in the OMOP CDM can then run it and repeat the process to also get the models for the same question. And the real value of this for prediction is the external validation. So if you have a model developed in one data set, say some claims data in the US, you really want to see how well it does around the world. So does it do well in slightly different patients in the US? Does it do well when you transport it to Europe or to Asia? And the nice thing is about the standardization, it makes external validation easy because the same code that I've used to extract my data, I can share with other people. So they can extract the data exactly the same way and run the model that I've developed. So we get a lot more open evidence from this. And the same thing happens if you're doing characterizations, if you're doing causal inference, you can do the code once and get it run across all the different data sites. It just gives you a lot more insight. Now for prediction, we've also standardized the process for developing and validating models. We wanted to have a very transparent a way of actually presenting a question. So we standardized the way you define a prediction question. And then we standardized the whole process of how you extract the data from the observational healthcare database, how you develop a model. And we have state-of-the-art classifiers as a library that you can pick as many as you want. And then we standardize the evaluation. It's important to have some consistency when you're evaluating a model with certain metrics to make sure that it's consistent. So if I develop a model once and I develop one 2 years later and someone else develops a model somewhere else, we want to be able to have something that's comparable. We want the same metrics. If people use different metrics, how do you compare the models? So we've standardized the evaluation metrics, and we also were able to standardize the way we do external validation and really scale up external validation. But standardization gets you part way, you also need data. Luckily in Odyssey, because we have a big collaboration, we all have the standardized format for the data. We also have a lot of data. So each dot here represents someone who's collaborating in the Odyssey network. We're a very open community. So if you can be someone who has data, you can be someone who has a research question, you can be someone who has computer science skills and they want to add in some latest machine learning algorithm. All these people are collaborating together. They're all represented currently by these dots. And across the world, we have a lot of patient data, all mapped to the OMOP CDM. Now, this meant that early into COVID, so this is the early part of 2020, we had millions of people being tested for the virus, millions of people being tested and being positive or being diagnosed with the virus, and hundreds of thousands of people being hospitalized for COVID-19. So across the network, we had very big data. Now, individually, one site by itself probably actually had pretty small data because we were very early into the pandemic. One single site is unlikely to have a lot of data, but collectively, we had a lot of data. And we can utilize that to be able to do analyses a lot quicker or sooner because we have this collective data. So just to give you an idea of how things actually went about in the 88 hours, I've got a little timeline of my 4 days of what I was involved in. So as I mentioned, I was involved in the prediction task. So I've highlighted in red here the things that I tended to focus on, but there was a big collaboration. So there was a lot of tasks as well that were necessary that I wasn't necessarily directly involved in. But hour 0 was actually at 1 a.m. in the US, and that was a bit too early for me to start. So I actually was sleeping through the opening presentation, but that was just telling people about the different areas of research we're interested in: characterization, causal inference, and the prediction. And it was telling people about how the whole 4 days were going to play out, and just preparing people, and people were picking what area they wanted to work on. That got recorded. So if you woke up like I did at 8 a.m., you could watch the recording and kind of just watch everything that happened up to that point in time, so you could catch up. When I woke up, I caught up, and I then presented the framework. So the standardizations that I mentioned earlier, we went through that in detail, and I showed everyone the open source packages that we have to run the study. So we tend to use R, is the language that we use as the front end. So I was showing them how we use the R package, and then people started to work together to create an analytical package that we put on GitHub. Anyone could contribute, and that was going to be used as our study package. At the same time, we had people developing phenotypes. So we needed to have a way of identifying COVID-19 in our data sets across the world. That's not actually a trivial thing to do. You have to do a lot of research and testing to make sure that you are identifying the people you want to identify, and to make sure that the definition transports across the world. So we had people making sure that each aspect of our prediction was valid. So we had people at ICU had to be identified, and different forms of COVID-19, and the tests needed to be identified. At the same time, we had people working on literature review. So we had people who were looking at existing models and seeing if there's anything that seemed to have promise that we could validate over the network. And so we had lots of people doing different tasks. By 10 p.m., I called it a night, and then I went to sleep. But because we have this global collaboration, it didn't stop. So we had started on the R package. I went to sleep, I woke up, and we had a finalized model package. So we had this package we put onto GitHub. It was available to the whole world, and anyone with data in the OMOP CDM could then apply it to their data to get models. So we had this developed by the time I woke up. We started testing it on some sites just to make sure there were no bugs and to make sure it ran as expected. And then once we were done with the testing on day 2, we started to develop these full models. So we had these large scale prediction models for each of the different severities of COVID-19 infection. And that's basically when I started calling it night again. So we had the models developed in a few sites around the world. I went to bed, and when I woke up, people had continued to work while I was sleeping, and they started to develop simple models. So as I mentioned, simple models would be a lot more applicable. People would be able to apply that around the world very readily. You don't have the barrier of how do you get this integrated into a healthcare system and such. So we started to develop simple models. Part of the reason we did the simple models is we had good performance for the full models. So we knew that there may be a chance that simple models would still do pretty well discrimination. And so we started to develop the simple models. It was basically 7 variables plus age and sex were all we were kind of using as predictors. We had a package for the simple models. Again, we put that onto GitHub. Anyone in the world with data in the OMOP CDM could run that to develop these simple models in their database. So we started to run these. We picked the model that seemed to do pretty well. So we looked at different data sites, but we could pick which one seemed to do well. And then we started to create validation packages of our full model and our simple model. So by day 3, we had defined our phenotypes, created a package, developed full models, developed simple models. And now we're at the stage where we were creating another package that we put onto GitHub. So it was our package, some R code, put onto GitHub that people could validate our models across the world again. So by the time I went to bed on day 3, people started to test that package to make sure that it was running on their data and there were no bugs. And when I woke up, people had finished the testing and we were able to start executing that. So day 4, we were able to execute the studies that were validating simple and full models across any data set that has mapped their data to any data set in the OMOP common data model. So we've completed our task of doing the prediction models. It didn't end there. We were still trying to get more data sites to do the external validation, just so we could get real good insight and feel for how well the models did. And actually, it was really fantastic results. We found that the models we developed consistently performed well across the world. So we had data sets in Australia, in Asia, in Europe, and the US, and they were all doing very well. So that was nice. We then created a website where people could interactively explore the models. So this is an app, a Shiny app it's called. So it's Shiny as a library in R, and it's a nice way of creating some friendly ways to interact with the results. We created one of a website. People could then go onto that website, view the models, view the performance, and explore everything. And then by 5 p.m. on day 4, we presented the results of the prediction. They presented, you saw everyone else's presentations on the causal inference and the characterization. So like I said, this was just one part of the whole study-a-thon, but we actually got a lot of insight about COVID-19 during these 88 hours. We also had people who had started writing up the paper. So people had actually been doing the introduction, the methods and results. So by day 4, not only had we developed 3 models, we had also got quite far towards writing a paper. So what was the output? So remember, bearing in mind this is in March 2020, so very early into the pandemic, we had already developed 3 simple models, and we had a risk calculator in a Shiny app. So this is the website if you're interested in viewing it. You can look at all the models we developed, look at the external validation, and you can actually look at the risk calculator that we developed. And then the risk calculator here, I've got a screenshot. So it looks at, you have to put in your age, you have to put in your sex, and then you've got these 7 history of illnesses: cancer, COPD, diabetes, heart disease, hyperlipidemia, and hypertension. And if you fill that out, it then gives you a bar chart that tells you your risk of hospitalization. So this is cover H, intensive care, which is cover I, and fertility, which is cover F. So this is your online app. Not only did we do an online app, we also created a friendly visualization. And this was all done because we have this diverse skill set. So there were people who were really good at doing the visualizations. There were people who were good at doing the Shiny apps. There were people who were good at doing the programming, the phenotyping. So it was really all this skill sets coming together meant that we were able to get so much accomplished in such a short time. So this visualization here is another way of how you can actually calculate your risk of hospitalization, intensive care, and fertility from COVID-19. And again, it has the same predictors, the medical history, age, and your sex. And then you can basically calculate a score. So this is how simple it is. You say what you have in your history, it gives you points. So if you had a history of cancer, you'd get +2 points. If you had a history of hypertension, you'd have +3, so you'd be at 5. Then you calculate your age. So if you're a 70 or 74, you've got 20, so you're at 25 now. And then you'd work out your sex. If you're a female, you then add on 43. And then that would give you a total score. And then you can convert that using the part 2 to get your calculated risk. So this is how friendly and simple the models ended up being. And as I mentioned, you can explore. You feel free to go to that website and explore, but the models actually had pretty good discriminative ability. The calibration wasn't fantastic. We'd have to recalibrate, but the discrimination was good. So what did we do after the 88 hours? We finalized the paper. So I mentioned that we got about 80% complete of the paper in the 4 days. We then continued over the next 2 weeks to finish that and submitted that. We didn't stop after the study-a-thon. So the study-a-thon was a great forcing function to make us do all this large-scale analysis for COVID-19. But it wasn't that at the end of 88 hours we stopped. We continued to collaborate all the time. So it's been ongoing since March. We still looked at more prediction questions that we could answer, and we've still been working on COVID-19 questions. We've also, as time progresses, we're getting more and more COVID-19 data. So March in 2020 was really early into the pandemic. So there wasn't that much data. I mean, we had big data because we had it over the network, but in general, the data per data site was pretty small. Now we're getting much bigger data per site. So we're actually able to maybe repeat the process using the bigger data we have available. I also needed to sleep. After an 8 a.m. to 10 p.m. day for a few times, I definitely was lacking on sleep, and coffee can only get you so far. So thank you for listening. If you have any questions, I'll be happy to answer.
Okay, thank you very much, Jenna. That was amazing. Thank you for showing us a window into your life for those three to four days. Thanks very much. And it just, I think, goes to show the power really of open science and open collaboration. And what you can achieve. I think. I mean, we did see the timeline for 3 to 4 days, but I think what we probably don't know about is how many years worth of work is behind the entire analysis pipeline, the analytics pipeline that that I think you all at the Observational Health Data Science Interactive Group used. So brilliant, thank you very much. So I think we are going to as well take a couple of questions now for Jenna in the interest of time only. I see 2 questions in the chat. I have another one pre-submitted, so perhaps we could start with that. So people are wondering, really just to get an idea of the sort of the scale of collaboration. So how many people were involved with this study-a-thon, Jenna? And connected with that question is, seeing as so much was riding on the analysis, the study packages, was there a process whereby that the analysis scripts were validated or cross-checked as well?
Yes, that's a great question. So in terms of the first, there were hundreds of people signed up. So it really, because it was a virtual event, it attracted a lot more people than our face-to-face study-a-thons have done in the past. So we had a lot of people for the prediction. I think we had probably around 20 people that were pretty active throughout the 4 days. So it was a lot of people hacking away. In terms of the kind of testing of things, so we have so what we were writing was really a skeleton using our prediction framework package, and that has a lot of test cases in it. So that is an R package that we put on GitHub, and we have a bunch of test cases just to make sure that it's running the way we want it. So that kind of does the check of that. For the actual individual study package, we tend to just call functions from the main kind of validated package, and we test it just to make sure it's running as expected. But a lot of the times you can see if there's an error because the data won't, like, you won't get anyone kind of being extracted, no data will be extracted, or there'd be some obvious bugs. So sometimes you do get bugs, and we kind of identify that from testing it. But the underlying things, so like the classifier metrics and anything really that's kind of really important, it's in the patient level prediction package, and that's got test cases and validated.
So which people can openly access and cross-check?
Yeah, that's all on, like, everything's on GitHub. So everything's very, very kind of transparent. We share like the protocol, we share the code, we share like everything online. And even the paper, we had that in the cloud where everyone was kind of like contributing to writing the paper.
So does that mean that someone from the school can become involved with something like this just by going on to getting in touch with you and some of the people who involved in this collaboration, and be involved if they wanted to join this collaboration on COVID analysis, and they had some data?
Yeah, we're always like, so you don't need data. So the collaboration is done so that if you have a skill set, like if you can do computer science and you can apply like some machine learning, we're happy. Like it's always great to have you. If you have data, that's fantastic as well. Like you can kind of map your data and then start running studies. If you're someone who has a research question, you're kind of like very like welcome to join and kind of say, here's a question I'd like answered. So different skill sets, different questions, different data, like everyone's kind of helps and kind of contributes. So yeah, we're very open. Generally, a good step would be to go onto the forum and start chatting with people, and then it won't take long before you start getting involved in running some of the studies or being involved in the studies.
Okay, I think we can close with just one question, but an important one. We've seen an abundance of patient level risk prediction models come out for COVID over the last few months and so on. Many of these, there's been a review of over 100 papers, and none of them were found to be bias-free, alarmingly. So one of the aspects of being, taking into account bias and so on is to do external validation. So were your models externally validated, and if so, on like how many data sets maybe, and from where?
Yeah, so we did external validation across a lot of data sites. So we had a site in Australia externally validated. We had some site in Korea externally validated. A couple of European data sites validated, and a lot of the US places. So I think it was probably around 8 or 9 different sites that ended up externally validating the model, the simple models that we developed, and the full models as well. That's where we found discrimination generally did pretty well, because age was the driving force of that, the kind of ranking. But the calibration was a little bit off. So we did find we had some calibration issues in some of the data sites. So you would have to recalibrate.
Great. Okay, thank you for those answers, Jenna, and thank you very much for your talk, even more so because we know that you were up at 4 a.m. in the morning where you are. So thank you very much. Hopefully you can go back to sleep after this. We will now move on straight to our next speaker. So our next talk is on data science for environmental sciences. So this talk was originally scheduled to be given by Professor Louise Slater from the School of Geography and Environment at the University of Oxford. Louise has unfortunately not been able to join us due to personal circumstances. However, we are very grateful to Katie Smith, who will present on her behalf. Katie is an expert on hydrology modeling and climate change. In addition to the work that she is presenting today that she did with Louise, Katie is also responsible for the European climate change project called Copernicus. So with that introduction, Katie, thank you very much for giving us time and joining us. It's over to you.
Thank you. I should clarify, I'm not in charge of Copernicus. Copernicus is massive. I'm just working as part of it. So yeah, I am here to talk on behalf of Louise, who sadly can't be here. I've done some work with her on a research paper, which was what she wanted to present. So I've stepped in for her to talk to you about this. It's been co-authored by a lot of other researchers all over the world, which is fantastic. And I'll give you a few examples as well of how we've used some of this research in my work at UKCEH. So the work that Louise has done with us is to release this paper on using R in hydrology. And it's really nice to see that Jenna's been talking to you already about how valuable R is as a resource for scientists. It's not just environmental science, although that's what I'm going to focus on today, and hydrology in particular, but in all aspects of science. So this paper really focused on the use of R in hydrology in particular, and all of the packages and resources that are available to hydrologists. We created this graphic to show you how R can be used at every single stage of a research workflow, right from setting up a repository, through retrieving hydrological data, cleaning up that data, tidying it, reformatting it, as Jenna said, is very important. If you're working in particular with lots of data sets from different sources, you need to get them into a consistent format. Extracting data in complicated formats as well from other sources, There are models, hydrology models, that are explicitly built within R. You can do some very complex statistical analyses using packages. And then my favorite part of Row research is how to visualize that data, plot it up with some really nice packages as well. And then finally, you can use a lot of R to actually publish your paper. You can publish it online, as Jen has already mentioned, using Shiny. You can use R Markdown and various other things to actually publish your paper. So why use R in environmental science and wider? The recent growth of R in hydrology has been driven by the fact that so much data is now publicly accessible. River flow and grid data reanalysis products are available, as well as remote sensing data from satellites, and then metadata catchment attributes as well. How big is the catchment? How urbanized is that catchment? And the land use, for example. There's been a massive increase in computational power. Even on a bog standard laptop now, you're able to do some quite serious data science, but we also have access to larger computers, often through our workplaces and universities as well. So there's a massive increase in computational power and the ability to use very large data sets. And with R as well, there's a very active and supportive community of users that I'll discuss in a bit. R it's based on the S language. It's designed for statistical computing, and in base R, there's a lot of inbuilt functions that you can use to carry out your statistical analyses. There are many techniques in hydrology, including linear and nonlinear modeling and statistical tests, time series analyses, and classification and clustering of results as well. So even within the base of R, as we call it, which is its bog standard language, programmed directly into the software that you download, you can do a lot of statistics straight off the shelf. But within hydrology, there's a lot of packages, and as Jenna was talking about, lots of our own research scientists around the world have been writing their own packages and publishing them for other people to use because they're really useful. And there are actually over 14,000 packages in R that are relevant to the hydrological science fields that have been released in the past 25 years. And these combine cutting edge technologies and tools and enable the seamless integration of lots of steps in that hydrological workflow, as I showed you in that diagram before. This is a little image here of the task view that a few of our co-authors have prepared. They've basically collated a website that shows you what all of these 14,000 packages look like and divided them into different areas, so you can find them easily and you can see what's actually available out there for hydrological research. And R has really democratized data science. It's really led now from the bottom up. It's completely open source. It's free to download. It's got really good documentation. There's loads of support on Stack Overflow. Anybody who's using R, indeed any programming language, knows how useful Stack Overflow is. There are a huge amount of packages, as I've said, and scripts also get shared amongst various communities as well. There are hosted instances of R. You can get RStudio in the cloud. So as Naomi was talking about, a lot of data services are too big to download. So you can use RStudio in the cloud to access those some of those large data sets directly without having to download them and do your research on there. And there are loads of platforms, Shiny platforms and things for people to explore analyses that others have done, freely available online. R is a really good way of doing reproducible research. So with the packages that most of us use, a lot of them are published on CRAN. Some of them have been through something called R OpenSci, which I'll discuss in a second, that's peer reviewed. And others have published code on GitHub as well, that's a bit less formal, but it means that if you've been using a package to do your analyses, you can cite that package in your journal and say, I use this package in this version of this package. And that makes your research much more reproducible. And then in GitHub example, if you actually publish your code on GitHub as well, then people can directly see exactly what you did. There are more and more journals actually themselves encouraging people to publish the code alongside with their analyses, so that can be part of the peer review process. Yeah, as I've mentioned, rOpenSci is a way of peer reviewing your software. So people have been writing packages in R, and they submit those to rOpenSci, so they get peer reviewed. And they can also be published in the Journal of Open Source Software, so that you know that the packages that you've picked up through rOpenSci or through CRAN and GitHub, some of them have been really well checked. Something I found quite recently, this is an example of a package that's been stored on GitHub, so you can download it and use it using the DevTools package, is the BBC. The BBC have released some of their plotting code, so they have shared their code, so you can upload your data. You can download their code and then use that on your own data to create a really high quality plot, just like they use in their in BBC Online. So that's really quite nice. R could be connected to lots of other languages. As in UKCEH, for example, there's different people use different languages for different purposes. And what's really nice about R is that you can embed bits of R code and attach it to bits of other code. So there's R to Pi, for example. You can spit things out of Python code and read it into R, or vice versa. And you can do the same with other languages, R, MATLAB, and Julia, and C++, for example. R has got a really nice integrated development environment. So it's got this IDE called RStudio, which is one of the most popular. There are others available, but RStudio seems to be the most popular. It's very like MATLAB, where it has lots of windows. You can see the code that you're writing. But as I found with if you just open base R, it's quite difficult to work out what's going on, what's been held in your environment. So if you've read in a data set in RStudio, you can see what data sets you've got in, what their dimensions, what class are they, and then you can click on it. You can have a quick look at it as a spreadsheet. If you want to plot a graph, you can preview that graph before you save it. And you can also see the command line and a script that you're working along side by side, and re-execute bits of your code as you go along without having to type it all in again. It's a really nice interface, and you can customize it yourself however you want to. R is also very good for big data and parallel computing. It's got packages that handle really large data sets, some environmental data sets, such as in the UKCEH, we've got butterfly data sets that are absolutely huge. But also a lot of the NetCDF time series of satellite data can be incredibly large as well. R can handle these quite nicely. And then it can also do parallel things. There's a package called Snowfall that I've used before, where it will split a job into little parallel sections. So rather than waiting for one of your CPUs to run everything in serial, you can split it up and say, I want to repeat this job in a very similar way 500 times. So please split that up and use 7 of the CPUs on my computer to do that. Application programming interfaces are also very useful. They allow you to directly access code from online. They're often provided with HTTPs, and one example that UKCEH produces, NRFA, the National River Flow Archive, is available directly online. So rather than manually going online, downloading data sets and then reading them into R, you can access them directly from R, and that's really nice because you can then iterate through the thousands of catchments that we've got without any manual steps in there. Another nice bit of work that's been done is on teaching environmental science through R. Some of the people that we've been working with work for Erstia in France, and they've developed the GR series of models, hydrological models. They're very, very simple lumped catchment models that represent the Earth's surface in catchments and route the water through, accounting for time delays and losses to evaporation. And they've developed this GR teaching site where you can run it through R, open it up through R, and it will pop up a little browser page, and you can then use that to interrogate the model, slide the parameters around, so change how much water is stored in the soil and how much water is stored in groundwater, for example, change how quick the water routes from one place to another and see what impact that has on the hydrograph. So it's really useful for teaching students how hydrology works, how hydrological models work. And yeah, it's a really nice way of explaining modelling. And there's a lot of community around R. There's lots of interaction and events. There's an extensive user community on social media. There's Twitter, hashtags. We've also got a Facebook hydrology group that I'm an admin for, so Hydrology in R, that's got thousands and thousands and hundreds of thousands of members, in fact. Stack Overflow has always got lots of discussion forums. There are mailing lists, as I've said. There's R OpenSci as well. I also have been convening and co-convening the short course at EGU, the European Geosciences Union, for a couple of years. We didn't run it this year because of COVID, and next year is going to be online as well, but we will run it next year. There's also an R consortium. There's an R Ladies Global, so talking about women in data science, there's an R Ladies in particular network for ladies who are using R. And there are lots of conferences exclusively to R as well, and help desks as well at scientific conferences. So I thought I'd tell you about a couple of examples of the way I use R in my research, and it's not just me that's contributed to a lot of this work. It's a lot of other people at UKCEH, and these are just a few of the wonderful ladies that I work with in my group. As we're having a nice recognition of women in data science, there are lots of us at UKCEH. So the first bit of work that I'd like to mention is my research on drought. So I did some work in the past 5 years on reconstructing historic flows. Observational flows in the UK are typically only available from about the 1960s onwards. That's when gauging sites tended to be installed and haven't really been moved since then. They've been maintained, but we have very little understanding of what happened before then. So yeah, this plot just shows you in red is sort of dry years, in blue is wet years, with the time along the bottom, and then catchments running roughly north to south along the y-axis. But yeah, what this shows you is how much data is missing. We've only really got quantitative data on 5 or 6 drought events since the 1960s. So if we're trying to look at what are the risks of drought events happening in the future, we've only got a few examples that we can draw on. So we had precipitation data and temperature data from the Met Office that went all the way back to 1891, and we thought, well, we can run this through a hydrological model to simulate historic flows. So we did all of this in R on the Jasmine supercomputer that's open to people, members of NERC, as well as other researchers. We ran 3 models, the GR in the AirGR suite that I mentioned earlier. We ran 500,000 model parameter sets to have a look at some of the uncertainties across 305 catchments. So that left us with over 450 million model runs to complete. So that's why we did this on the supercomputer in Jasmine. But it was all coded up in R, and it ran really nicely while I was away for Christmas. And this is what it gave us. It completed that data set running for the whole country all the way back to 1891, and gave us a lot more drought events that we can then interrogate for our research. Some of these drought events, such as those, the one in the 1920s and in the 1940s, really covered the whole of the country. And then there's also the drought event that happened in the turn of the century that spanned over a decade. That's a really unique event as well. So it's been really valuable for us to have that information in our drought research. And then finally, as I've said, you can use R all the way through your workflow. So this was quite a big data set that we wanted to release for people to look at. And we wanted people to be able to interrogate that data set in quite a lot of detail, no matter where they were in the country and what their interests were. So we built some Shiny apps that are available online. They're fully interactive. So rather than having to download the full data set and try and plot their own graphs, people can scroll around the Shiny app and have a look at what they're interested in. So they're able to look at the full time series and then drill down and zoom in on in particular time steps within that. So it's not just for our pure research that we use R, we also use R for our operational services. An example of that is every single month we produce a hydrological summary of the status of flows in the UK, and then we also produce an outlook that's a forecast of flows over the coming season. And for the hydrological summary, we get this data into the NRFA. It's tidied up a bit with SQL, but then it's all plotted up in R. But for the hydrological outlook that we run, several of the methods that we use to run are all run in R and plotted up in R as well. So those forecasts are done operationally in parallel in R. And it's not just pure environmental science that we use R for as well. I'm currently working on more of a social science piece of work, looking at the ways in which our country communicates drought risk, be it from DEFRA or the Environment Agency, or from water providers and water companies. And so we've been doing a survey on how people perceive those drought risk communications, who do they trust, and we've been using R to analyse a lot of those results. So to conclude, R is an open source programming language. It's really quite central now in hydrological research as well as in other areas of environmental science. And it's also being used in operational practice. It's a very flexible language. It's very easy to use. It's very similar to other languages, particularly MATLAB and things are quite similar. So it's an easy way to jump from one to another if you've had previous training in other languages. There's a lot of tools available. It has a huge amount of code in repositories, and that's going to become more and more standard practice to submit your code to repositories and have that code reviewed along with a manuscript. And we think that R is going to really continue to advance and play an increased role in environmental science and in hydrology. So thank you. Do I have any questions?
Thank you, Katie. Thanks very much. I think that was really interesting to see all of that data going back to the 1800s. And also, I mean, I think over the years, the data science community, computer science community has moved, as well as the statistics community, I should say, towards pervasive use of R. So I'm sure the audience today will have appreciated this in-depth view of of how to use R in general, and also in particular for environmental sciences. At least from speaking for myself, I had no idea how well developed the environment is with so many packages just for one field. So fascinating. Thank you so much. Again, we have time, maybe this time just for one or two questions to take from the audience for Katie. And while we're waiting for people's questions, I guess one question that I can pose, Katie, from some of the pre-submitted ones is so all of these data that some of this data goes back a century or more. Has this in any way brought out any signal as such on climate change? So I know this is environmental data, but have you picked up on anything that tells us anything about how things are changing in our climate as well?
Yeah, so there's a huge amount of research being done on trends in environmental science data sets. And definitely there's a particular trend that I've seen from the Met Office that have picked up on temperature. Temperature is very steadily rising. And so when they release their forecasts each month that we then use for the outlook, they have to tweak the model to account for that change. They compare their results to a baseline of 1981 to 2010, and things are changing very, very quickly. So they do tweak that accordingly because they think that the most recent 10 years have been really warm. But yeah, there's a huge amount of science involved. It's more difficult in hydrology because of the human influences as well. So abstractions and building of reservoirs have changed, and urbanization have changed the way that water moves in the landscape. So it's very difficult to model those things, and it's very difficult to work out which aspects of hydrology trends are related to human influence and which trends are related to the actual changes in the climate, which is a perpetual problem in hydrology.
Exactly. I think there's one more question that we can take, and I think it's quite a good one as well. So, is there scope to combine data from drought effects with health data to look at obviously the effects on health, on human health?
Yeah, it's not something that we've been doing at UKCEH as much as I think some of the other universities have been researching this. I know that droughts are very correlated with heat waves, and I do know some researchers at Loughborough have been looking at all around the world, in fact, what impact heat waves have on fatalities. So yes, there's a lot of research going on that, but yeah, not so much by us.
Right. And I did say that was going to be the last question, but I remembered there's been a lot of demand for this one question for your talk, and that was about and this, I have to warn you, it's not particularly hydrology or data centric essentially, but the question in many people's mind about is there a link between pandemics and climate change, and is climate change going forwards likely to cause more pandemics to happen?
I wouldn't call myself an expert on that at all. I think, yeah, it's hard to say. I think as climates change, the way societies are evolving have changed. And I think watching a lot of David Attenborough's programs, it's been quite apparent that the way societies are now encroaching upon biodiversity and upon other parts of the environment and really impacting pandemics coming out of those environments, and that jump from animals to humans is becoming quite dangerous. So yeah, I wouldn't say I'm an expert, but I would encourage you to go and watch some of the programs that David Attenborough has released in the past few weeks.
Okay, yes, we were getting some interesting comments from the audience on this one as well, and I think this is a conversation that we can dedicate a whole other day and a session to. But thank you so very much, Katie. Thank you to all our speakers. I hope everybody has enjoyed just as much as I have. We will now move over to our panel discussion.
Yep. Hello, Sarah.
So,
thank you very much. So shall I start?
Yes. Yes, sure. So over to you, Maria, for the panel discussion.
Okay, sorry. Okay, so you're sorry. Yeah, thank you very much, Sarah. So hello, everybody. Yeah, my name is Maria Sanchez, Senior Research Associate in Epidemiology and Medical Statistics at University of Oxford, and I'm going to be the moderator of this panel discussion on women in data science. So it is really a pleasure to be the moderator. So before starting, I would like to ask to the panelists to switch your cameras to cameras on, please. Yeah. Okay, thank you. So, well, I'm going to do a brief introduction of our amazing panelists. So we have Professor Naomi Allen, so Professor of Epidemiology at the Nuffield Department of Population Health and Chief Scientist for UK Biobank. Her research focuses on the use of big data to understand the role of diet and hormones of cancer risks. So we have also Gemma Rips, Associated Director in the Observational Health Data Analytics Group at Janssen Research and Development. Her main role is to develop open source and user-friendly tools for risk estimation model using big data. And we have, finally, we have Antonella Del Mestri. Antonella is a senior health data scientist at University of Oxford. She is responsible for the automation of data engineering, data mining, and advanced curation of real-world health data routinely collected by general practitioners, hospitals, and registries. registries, sorry, among many other things. So thank you really very much for joining us today. So first, I'm going to start with a question to all of the panelists, and then I'm going to take some of the questions from the audience. So if you have any question for the panel, please use the chat box on the right, to share your question. But if you don't have any question at all, so but you see one of the chat that you would like to be asked, so please use your thumb ups icon next to the message box. So there we go. So I would like you, our panelists, to tell us a little bit about your journey. So how you made this career choice, and what do you think was the biggest challenges as a woman, if any? So Jenna, would you like to start?
Yeah, sure. So I basically did maths at university because it was the subject I enjoyed the most when I was growing up. And then I found that I really liked applied maths. So I kind of moved towards the mathematical biology. That got me interested in kind of the more computing kind of side of things. So I ended up switching to computer science, applying for a PhD in computer science. I was starved of data. Actually, when I was doing my my master's, I had no data. So we were trying to model the immune system, and I was trying to like fit kind of like things through optimization, and there was no data really to use. So when my supervisor, my PhD, said we've got a project where you have this big observational healthcare data, THIN it's called. It's like kind of similar to CPRD as well, which is kind of used a lot in the UK. He was like, we've got this and you can use it to basically just do computer science stuff on it, and it's pretty open, and just like we want to get some value out of it. I was like, great, I'll have a go at that. So then I spent my PhD using that to try and identify side effects, and that got me into using that data for prediction. And then that kind of got me spotted by my current boss, and he was like, oh, would you like to make things more generalizable and just do prediction in general with this data, and kind of more data in that kind of format, so the US claims, and then kind of got into Odyssey as well. So now we have data all over the world, and it's pretty exciting. Challenges, I don't know if I really had any challenges to date, but I think that I've been quite lucky to have good leaders. So especially where I am now, the people in charge are very kind of considerate of things, and it's a really great atmosphere. I'm in a team where there's a lot of women, so I think it's about 50-50 in the team, women to men. So I feel like that pretty other people have had the challenges, and I'm benefiting from them kind of coming up with solutions. So and I think also in the company there's a lot of visible leaders, women leaders, that's kind of helped as well. So in the group I'm in, the actual kind of one of the more senior leaders is a female. So I think that she has obviously a good perspective, and she makes things a lot easier for us.
Great. So who is next? Antonella, you want to go next?
Sure. Thank you, Maria. So my journey has been a bit bumpy. So I started as a computer scientist, and I spent like 15 years developing software and firmware for electronic devices. I never had a female colleague, so it was pretty lonely in a way, and I definitely had the intruder syndrome, because I just felt I didn't belong there. And then I thought I needed a change, and I started a PhD in information and communication technologies. And at the same time, I started searching for a different job. And interestingly, I couldn't find absolutely anything in Italy, because I'm from there. So after trying for a few months, I published my CV on a UK website. And in 2 weeks, I had a surprising change of life. So I had 2 contracts, one with Oxford University, which I joined in 2006 at the Clinical Trial Service Unit, and another contract with the TomTom in Amsterdam, the satellite navigator company. And I decided to come to Oxford because I could have finished my PhD. That job for me was, let's say, easy. So I have been able to work full-time and do my PhD in my free time, meaning nights and the weekends and things. So yes, lots of challenges on my side, and I still have many. And one is the difficulty of finding mentors that have my type of background and can advise in how to progress. So I had always to invent myself in a way, because in the data science, I'm at the beginning of the chain. So I'm in the database management and data quality, data curation, which is in a way taken a bit for granted, maybe, because all the attention is in the second part of the science. Yeah, so that is my personal experience so far.
Okay, thank you very much, Antonella. So listen, that different challenge. So different people, different experience. So that's good. So maybe we born in a Rome country, Antonella.
Absolutely. Absolutely.
Okay, Naomi, you next.
Yeah, I'd say I had a very difficult beginning. So I had absolutely no idea what I wanted to do at university. As a teenager, I had these vague notions I wanted to save the world, like everybody does. And I did environmental science, which I absolutely hated, apart from one module on public health and epidemiology. That was the only thing that really floated my boat. And when I finished university, I was unemployed. I had a string of terrible jobs. And after a couple of years of that, I thought, oh my God, I've really got to do something to change my life. And I thought, what's the only thing I'm really interested in? It's public health and it's population health. And so I did a master's in London in epidemiology, and that I haven't looked back since then. And that was in kind of my late 20s. And then I did, the usual route of a PhD and so on. And that was really the start for me. And I think having that couple of years of being in limbo, floating around, having no idea what I really wanted to do, gave me a real fire in my belly and impetus that actually once you've found something that you're passionate about, you work really hard and you stick with it, and you can have a career that you're passionate about, and that is entirely possible. So yeah, I think the key is really finding something that you're really passionate about and sticking forward. And I think the other main challenge is obviously the age-old one of trying to juggle career with looking after a family, and that's a perennial struggle that we all deal with. And I think things have changed a lot, certainly where I work in our department over the last 10 years or so, with flexible working. And even now that we're all working from home, for some people it's much harder. They've got young kids around their legs, but for other people it has huge advantages because you can work much more flexibly and you can work the hours that works for you. So I think there's a real opportunity going forward in making the work fit around our other priorities in our life now.
Okay, thank you very much, Noel. So yeah, I think I agree with you. So I think we yeah, it has been done a lot, but I think we need to do more. So it's something like, yeah, something that we had to, yeah, to work, not just us. Yeah, I think everyone. So yeah, okay, but thank you. And now I'm going to take some questions from the audience. So I see that there is just one, but it's for Diana and Naomi. So what advice would you give early career public health researchers who want to make a transition to a more data science focused career?
Shall I start? Well, there are loads of online courses you can do, many of which are for free. R is open source, so you can download it and have a play. I'd encourage you to get involved in these hackathons that was talked about earlier. Even if you're not a super whiz at R or data analytics, it's a really good way to muck in and get involved and learn from others. And ask lots of questions from people, and because people around you will absolutely help you in that endeavor. So do as much as you can online and hackathons, and then think about doing a PhD or talking to people in the data science field as to how you can make that transition. Jenna, I don't know if you have other pearls of wisdom.
Yeah, no, I think you covered that perfectly. Yeah, I would say just basically get some experience if you can with hacking with the data, like just having data and playing with it in something like R or Python or however you feel comfortable gets you a lot of insight. And then yeah, the online resources now are amazing. There's so many online courses. You can like Book of Odyssey, if you're interested, has a lot of information about causal inference or prediction in the area, and you can feel free to hack around with the code we have. But yeah, so I think and then it was mentioned, if you really get serious, then a PhD as well could be something of interest. If you haven't really done a PhD and you're kind of fed up of it, a PhD could be a good way as well.
Okay, thank you. So there is it is not a question, I think it's a comment. So it's just to say thanks to Antonella and Naomi for breaking the glass ceiling of women in data, so that now there are much more options and at least some roles models for younger women. So thank you. So there is a very good question here. So yeah, do you think does gender play a role in how data scientists approach their work and the topics they choose? For example, improving lives versus creating destructive weapons? So yeah, I think that's a very good question. So Antonella, so do you want to start with pick one?
Yes, sure. So in my experience, I don't think the gender plays a role in how people model or program. I think what plays a big role in that is your background. So which type of degree do you have? Because now programming is used everywhere, and not all the disciplines teach people how to program. So many people sort of learn as they go ahead, focusing on the topic, the objective. So I think that could play a role, not the gender, but the gender I think play a role in the in the topic people get passionate about. For example, I know more men being very excited in developing video games than women. This doesn't mean that many women don't like developing video games, but on average, I think that could be more a male interest, while maybe women are more interested in developing something useful, you know, that can make a difference, not just entertaining. But this is just my thoughts. So I'm curious to hear the other speakers. What do they think?
Yeah, I think I have a similar kind of opinion. I think that it's more kind of the way you've been taught that kind of dictates like your approach. I mean, maybe there's some creative elements to kind of data science that maybe there might be a difference in average on kind of how gender would kind of interact with that. But a lot of the times it's your background. If you someone who's kind of got the epidemiology background, someone who's coming from computer science, I think that kind of has like a different approaches. But yeah, I do think that there might be a different application. So I know when I was a researcher in Nottingham, we were trying to get more females into computer science. One of the ways we tried to do that is by advertising that you can do computer science for healthcare. So we realized that women tend to prefer healthcare as an application, and we kind of try to use that. So I do think there is like a bias in where you actually would apply data science.
If I could just come in here, I think the way that gender does play a difference is often in team dynamics. So obviously, you know, science is very much a collaborative endeavor, and certainly in my experience, teams led by women often work very differently to teams led by men. And I think having a team dynamic which is, you know, kind of collaborative, and where women have been through the experience of having to get back to pick up their kids from play school and not having this kind of 24-hour, 24/7 working culture, is and much more kind of a bit more of a nurturing environment with teams led by women compared with men. I know that's a gross generalization, but it does tend to be largely true. I think that is starting to change how productive teams are and how people interact with each other across different teams as well.
Okay, very interesting. Thank you very much. So we have, I think, one question. Let me see. So it's one comment from Sarah. So that is one approach for women to progress is for women to support and mentor women. However, there is some research suggestion that females mentor by females progress less than females mentor by men. What do you think? So Naomi,
yeah, Maria,
yes.
sorry, these are just some questions that are coming in from the audience, so I'm just
okay, sorry.
that for you. Yeah, go ahead.
Okay. So,
well, I don't know about that. I don't know what the evidence is to back that up. You know, we have a very good mentoring scheme in the Medical Sciences Division and in the Nuffield Department of Population Health. And some people specifically ask to be mentored by men or women. That's their choice and that's fine. But I don't know personally of any evidence to suggest that if you're mentored by a woman that your progress is less. I think that's much more anecdotal. So I'd like to see some evidence to back that up.
Okay. Okay, so a question from Nina. So she said, how would you recommend making the transition from pure mathematics to more applied data science? And at what point do you recommend making the transition? University, master, PhD, etc. So Naomi, do you want to start this one?
That's a very good question. I think being able to do a PhD, I think university actually is it's still a broad playing field. You're not closing too many doors, but it's really at the master's and PhD level where you're starting to get very specialized. So, I mean, just in my own field, you can't do an undergraduate degree in epidemiology. You have to start at an MSc or PhD level. So it really depends on which area you're in. But I would say MSc, PhD stage is really where you need to start focusing about what it is you're really passionate about. Others may have different views.
Yeah, so I think like with the pure maths background, like it will give you potentially, I mean, it depends what area of pure maths, but it could give you a big advantage, I think, actually for kind of data science, because you will generally kind of like be able to understand all the underlying theories, is what I found. So I think like doing like a pure maths degree does set you up a good foundation for data science. I would probably recommend that you would transition at least for like a master or PhD personally, just because then you'll get the hands-on experience as well. So you'll have like kind of the background in the theory, and you also have the hands-on experience. But I think that a lot of times when we're trying to recruit people in data science, having like a maths background doesn't rule you out. Like if you have a PhD in a pure maths background, that wouldn't necessarily rule you out. You're still kind of like if you've got some experience with data science as well, then you can pretty get into the data science field still pretty easily.
Antonella, would you like to say something?
Yes, I agree with Jenna and Naomi. Really, in my experience, when you have a very strong logical background that maths give you is really the best foundation. You know, you can build up anything you need, and you just need to be able to apply your logic to some real problems. So a master, a PhD is a great way to progress and also have, you know, that recognition of the knowledge you are building up for your future career.
Okay, thank you. So I think we have just 5 minutes left, so time flies. So thank you very much. So I would like to ask each of the panelists, what would your best tip be to your younger self? Who would we like to start? Jenna, for example?
It's a tough one. Maybe to kind of be more assertive when I was younger. I guess like I feel like now I'm maybe getting a bit more assertive, and I think like that's kind of something that like I've developed. But as a kid, I was pretty shy and not assertive, and that maybe was one of my challenges growing up.
Okay. Thank you. Naomi?
Yeah, that is a really tough one. I think my advice would be don't think you can do it all, because you can't. It's impossible. And if you have those, you know, perfect expectations, you will fail and just disappoint yourself. So have realistic goals and don't eat all the elephant at once, and just chip away at things that are realistic and obtainable, and in the end you'll get there.
Thank you.
Yeah, I would say be confident, look at the results you have achieved, not at what other people may want from you. Try to keep away from your mind the stereotyping and other people's expectations, and look at your strengths and build up on them.
Okay, thank you very much. So, well, now I'm not want to take any more questions from the audience. So, because we are in the end of the panel discussion, so I would like to thank our panelists for a really interesting and fascinating discussion. Thank you also to our audience for their very interesting question and comments. So I hope you enjoyed. And now I will hand it back to Sarah for the closing remarks. Thank you very much.
Right. Thank you, Maria. Thank you, panelists. I think that was a really engaging and thought-provoking discussion. Just making sure I'm not muted. That's one of my worst nightmares. So all looks good. Yes. So yeah, thank you so much. I think our hope is for the conversation to just keep moving in positive directions. And with that, I have to say that we have come to an end of our program for today. I hope you've all enjoyed. As you know, this was the first event of WIDS Oxford, and we want to make things better for the future of WIDS and to expand this network. So we would love to hear your thoughts and ideas on that. So what that means basically is that this will be followed up by a feedback form. Do let us know how you felt things went, and give us your ideas for future events. We do have a mailing list, and some of you have already opted in. If not, and if you would like to join the mailing list, please email us at wids-ndoms, NDORMS, at ox.ac.uk. And that way we can keep you informed of future events at both WIDS Oxford and WIDS Global. And also for you to keep in touch with us. So with that, all that remains for me to do is to thank everybody, including our speakers, our panelists, and most importantly, our audience, for joining us for this event today. We do have a follow-up session now, which will be led by Daniel, and that is about the planning. Actually, it's for anybody who wants to get involved with WIDS Oxford and help us plan things for 2021, including a datathon. Do please stay back for that session if you're interested. And for everybody else, otherwise, thank you so much once again. Have a good rest of the day, and good luck. Thank you.